What exactly are vesicles? Before we dive into the energetics per se, let's just give people a basic idea of some of the key pieces of infrastructure within the synapse. Let's start with vesicles. You mentioned those. What exactly are vesicles?
So synaptic vesicles are bounded by a phospholipid membrane, a bilayer. This is, as you said, it's fat because it has a carbon chain that's usually, maybe a dozen to 20 carbons long, depending on sort of now details, with a charged head group, the top of it. So these things will form this little container that's bounded by something very hydrophobic that doesn't like water and forms a barrier that you then can package in whatever you want. But to package things in the vesicle, you need specialized machinery. So living on the surface of the vesicle are many other types of proteins. In this case, highly relevant to the conversation we're going to have is you need the thing that's going to allow you to put neurotransmitter in it. These are transporters. There's a different flavor of transporter for every different flavor of neurotransmitter. There's one that's specialized for glutamate. There's another one that's specialized for GABA, the major inhibitory neurotransmitter. Another one for acetylcholine, the major neurotransmitter, for example, for allowing muscles to contract, but also present in the brain. Another specialized one for dopamine. And so each one, they're specialized machinery. They're kind of all similar to each other, but they have their own little specialty of why they work well for one type of molecule, not another. A vesicle that's ready to go might have as much as a few thousand molecules of, say, the neurotransmitter in it. Now, the concentration of neurotransmitter in the outside of the vesicle is actually on the order of sort of, let's say, millimolar, for example. But inside the vesicle, once you packaged a few thousand copies, it's probably more like 100 millimolar. So that's a giant gradient, and it takes energy to overcome and build that gradient.
Because if you concentrate something in a little bubble, it will naturally want to go back out down that concentration gradient. So you have to pump, you have to use energy to keep things concentrated.
And one of the big, I would say, discoveries of the last 30 years, say, is the beginning to understand what are the machineries that do this pumping and what do they rely on. And you might have said, oh, well, you can make a pump that's powered directly by something like ATP, but in fact, that's not the way it works. Instead, all synaptic vesicles rely on the action of another pump, a proton pump. That is actually called a V-type ATP, is V standing for vesicle or vesicular. And those live in more places than just synaptic vesicles, but their big thing is they use up ATP to pump protons. And so it turns out the way you do all of neurotransmitter pumping into vesicles is sort of a coupling of the energy you get from pumping protons and creating that gradient first. And that ends up being the power to put in neurotransmitter.
So vesicles contain neurotransmitters, which are key components of synapses used for communication purposes. Vesicles are not merely little bubbles of fat, little bags of fat that contain chemicals. They have embedded in their membrane, just like the main cell membrane of the cell, a bunch of proteins that are important for actually keeping the vesicles filled with whatever chemical a particular vesicle has.
As well, of course, is the whole business end of how do you know when to fuse with the membrane to release your content.